can see.
Yeah.
Okay, perfect. So this is where we were in the last class, right? We started with the stages of sexual reproduction in plants. And we also have Nida joining in. Good evening, Nida. How are you? Am I audible and visible to you?
Yes.
Yes. Okay, so Nida and Aisha, this is where we were, right? In the last class, we started with the stages of sexual reproduction. Any doubts you both have? And you both will be having a test, a 30 minutes, it will just be a half an hour test after the class, okay?
Okay.
Okay. And what I'll teach today will not be included in the class. So it will be just the basics. And the motive of the test will be to assess your subjective answers writing capability, because it's an important part of your boards and school examinations that you write good subjective answers. You know, Aisha has this problem, but it was bigger one in the last year. I think in this year she's doing better, right? You can now answer better, like pinpointed, not beating around the bush. But today's exam is just to help you with that. Okay, so it will be a short 20 marks exam with total 8 questions that you have to do in 30 minutes. Okay,
Wait, is it like 2 marks, 3 marks, this kinds of questions?
is it what?
Like the questions will carry marks. Is it like 2 questions will be of 2 marks and 3 marks like that?
4 questions carrying 2 marks and 4 questions carrying 3 marks. Okay, so read the question and everything we have discussed in the class already. So read it and write it, and then you have to submit that. Click picture and send it on the official number, the WhatsApp number from which you get the class invitations links of Learnivio. So just write your name, write my name and your batch and send it there after you finish. So it will eventually come to me and I'll check it and discuss with you in the next class. Okay, Aisha and Nida?
Okay.
That's after the class. Okay, so
Can I say you one thing? Like if you are going to say the performance in the next class, so sir, at that time also join early because I don't want anyone to hear about.
sorry, I could not understand you, Aisha. What?
Next class, I want to join early and I want that at the time no one will be there so that at 2-3 minutes you can say and you can comment all the stuff about my feed because I really don't want anyone to listen about me.
Okay, so I'll not talk like about your performance. I'll just in general discuss the paper and what generally should have been written in the answers. And I'll, if you wish to discuss something with me, we'll do that. Okay, it will be general. It will not be pinpointed to someone.
No, it's not like that. I never mind that.
Okay, so let's start with today's class. You understood, Nida, about the test?
Yes.
Yeah, but that's after that. Let's focus on the class first. So we already started with the stages of sexual reproduction, and the first stage that we already know is pollination, which is basically where the transfer of the gametes takes place from the anther to the stigma of a flower. Okay, and so this part basically was about transfer of gametes, right? And we did that part. And in pollination, so I'll give you the brief recap today, both of you. So pollination is basically, as we know, it's the transfer of pollen grains from the anther to the stigma. Now this can happen from the anther of the same flower to the stigma of the same flower, which is called autogamy, the first case, or self-pollination, which is within the flower. It can also happen between the anther of one flower and the stigma of another flower, but both of them are present on the same plant. So that is called geitonogamy, between two flowers, but within the same plant, like on the same plant. And then we did xenogamy, which is a cross pollination, which is between different flowers present on different plants. So an anther of one flower transfers the pollen to the stigma of another flower, which are completely unrelated, like in terms of genetics. Okay. We also have Nora joining in. Let her join the class. And I ask you all to go back and find examples that on the basis of which I had to discuss something in today's class. So did you find examples, Aisha, Nida? And good evening, Nora. Am I audible and visible to you, Nora?
Yes, yes.
Yes. So did you go back and find the examples for autogamy, geitonogamy, and xenogamy, which I gave you as a homework, all three of you? No? No one did the homework. That's not good, okay? See, when I give something as a homework or an in-class assignment, that is because it will spark the discussion in the next class. It actually is for a reason. Now, since none of you have done it, anyone has done it? Aisha, you have done it?
Yeah.
You've done
I
it?
wrote it. Actually, I took my book and from there I did little bit research and I didn't do everything for myself. So I wrote it in my book.
Did you find examples of autogamy, geitonogamy, and xenogamy plants?
Yes, sir. Yes, sir.
Good. So tell me the examples.
Just 1 minute. Let me take my book. 1 minute.
Yes, please. Nida and Nora, have you done the assignment, the homework? No?
Yes, I did. Just a minute.
Okay, please find your copies and come up with answers. Let me know when you both are back. What about you, Nida?
Hello?
Yes, Aisha, you're audible.
Yes, sir. Yeah, for autogamy, sunflower, peas, then what we are saying, it's orchids, and then tridex. And for deuteronogamy, it's corn, and that's only the thing which I got. And
Corn,
for
right?
yeah, corn, yeah,
And for xenogamy?
and for xenogamy, I got onions, broccoli, then spinach, and then grasses and olives.
You got onion,
Olives,
broccoli,
yeah,
okay, olives,
yeah.
okay. And
Okay, by the way, I'm confused. I wanted to ask you, is
yes,
grass also xenogamy?
grasses, yes, grass do produce flowers, and yes, grass can
So
do
it means trees also, right? Trees will also come.
peas,
Trees, paid.
trees.
Yeah.
So trees, like which tree you are talking about? If you're talking about flowering trees, then they are.
I'm talking about flowering trees on leaves. Some trees are there, like, some mini trees are there, like that what you're saying. Champa, I don't know what to say it in English.
Yeah, it is called champa.
Yeah.
Yeah, it is called champa only.
Okay.
I think,
Is it that flower is also coming in that category?
yes,
Like it's a small tree, right?
yes, they're smaller. They're smaller trees. Let's also go to Nora. She also has some examples. Nora, what are your examples for autogamy?
Um, for autogamy, sunflowers, orchids, and peas.
Same. Okay. And for geitonogamy,
Corn.
what are your sources? Like, are both of you are searching the same source somehow? And xenogamy, Nora?
Onions, broccoli, spinach.
Yes. Now, where did you find this from?
Sir, it's
Yes.
just a common example which you can find it everywhere.
Yeah, right. So what I wanted to see was that both of you have figured out P, the autogamous, to do autogamy. And do you know that uh, P comes in chasmogamous plants or cleistogamous plants?
Cleistogamous.
Yes, so P is a cleistogamous flower. Okay, and if P has closed flowers, the female part is not exposed. It do not open. The flowers do not open. And for that reason, if question comes that what kind of pollination does cleistogamous plants allow, the only kind of pollination that cleistogamous plants like pea can allow is autogamy. Do you understand? No geitonogamy, no xenogamy. Is it clear?
Yes.
So write down cleistogamous flowers can only undergo self-pollination.
Hey, can I ask one thing?
Yes.
Actually, I was having a doubt and I wrote it in my notebook but because I wasn't confirmed what that answer is. Papaya geitonogamy? Like,
Yes,
I'm so confused with it.
papaya
It is.
geitonogamy because in papaya there are both papaya are dioecious.
Yeah, they
Papaya.
have both. Yeah.
So you know papaya, write down, you can write down papaya. It's all the dioecious plants can only do geitonogamy because either like papaya is monosexual for flowers, but both type of flowers can be present. So papaya and other plants which produce monosexual flowers can undergo geitonogamy or xenogamy because in one flower only one type of structure is present, either male or female. So if either male or female is present, so if a question comes that a particular plant produces monosexual flowers, then what kind of pollination it can allow? If it is monosexual, then it can either allow geitonogamy or xenogamy. It cannot allow autogamy, right?
Yes, sir.
Yeah. So any kind of a question comes on this basis, can you answer it now? Aishanida and Nora?
Yes, sir.
Okay. And in today's class, I told you that we'll be starting with double fertilization, embryo sac, the structure, and what is triple fission. Did you go back and also search for these terms? What do they mean? Anyone did this?
Yes, sir, I did it.
Okay, so let's start with the first topic, that is double fertilization. And basically this is the second stage of sexual reproduction in flowering plants. The first was pollination. Second is fertilization and formation of zygote. And what happens after the pollen has landed on the stigma? There is something called pollen pistil interaction. So let's go to that before double fertilization. Write down. Do not write double fertilization. Write down first the second stage, fertilization and formation of zygote. So you understand that now when the pollen has landed on the stigma, it is going to do what it is made to do, right? It has to go to the egg cell and fertilize. But before that, let's also discuss the structure of pollen. I think we have not discussed. Yes, we have discussed the structure of pollen. Can you see in a pollen grain, as I've already told you, there is a generative cell and a vegetative cell, right? Everything about this pollen grain is clear to you, which we did in the last class?
Yes, sir.
Yes. Now this generative cell, just before the formation of pollen tube, it divides into two, okay, giving rise to two male nucleus. So when pollen tube is forming, and which cell is responsible for forming the pollen tube? The vegetative cell. So vegetative cell gives rise to the pollen tube and generative cell gives rise to male gametes. Now there are two male gametes. Just before the pollen tube starts forming, the generative cell divides into two, forming two nucleus. And write down, when the pollen lands on the stigma, when the pollen lands on the stigma, the first event that takes place is known as pollen pistil interaction. Okay. So what happens is, if this is the stigma and pollen just lands here, so this is the pollen that has landed. Now this pollen will start interacting with the stigma. So at the surface where the stigma and the pollen meets, there is a biochemical reaction that takes place. Write down. Biochemical reaction. Okay. And this helps the stigma to recognize the pollen, whether the pollen grain is of the same species or not. Make sense? Because if a stigma is exposed to the environment, any species pollen, any pollen can just come and land on the stigma, right? It's an open platform and any pollen can land. It's not necessary that all the time the pollen of the same species will come. Yes or no? Do you understand?
Yes.
So it's very important for the plant, for the female structure, stigma, to first figure out whether this pollen is of the same species or not. So write down, the pollen pistil interaction leads to identification of the pollen belonging to the same species. Belonging to same species. Okay. And this decides the compatibility. Once the compatibility is there and this interaction is successful, then this pollen grain is allowed to form a pollen tube. Okay. Is it clear? So this is pollen tube. Is it clear? And in a pollen tube, if I draw it like this, this is a developing pollen tube. Then inside the pollen tube, there will be nucleus, right? How many nucleus will now be there total in the pollen grain? One will be the vegetative nucleus, and there will be two nucleus from the generative cell. Remember, I told you that just before the pollen tube is formed, the generative cell divides into two, right? So if I zoom in this part here, make it like this, then let's say this is the stigma. Pollen. This pollen is forming a tube. This is the developing tube. Okay. Inside the tube, there will be nucleus. There will be three nucleus. There will be a first nucleus which will be leading, and this will be the vegetative nucleus. Correct? You understood, everyone? And behind the vegetative nucleus, yes, you're saying something, Aisha? There will be.
You asked is it clear, so I was going to say yes.
Okay, and behind the vegetative nucleus, there will be two more nucleus, which will be the male nucleus. How many nucleus in total in the pollen grain, in the developing pollen tube?
3.
3 nucleus. So if the question comes that what are the first two leading nucleus in a pollen tube. So what will be the first two nucleus? A vegetative and a male nucleus, right? You understand? This is a neat
Yes.
question. Write it down. The first two leading nucleus in a pollen tube is a vegetative nucleus and a male nucleus. And if the question comes, what are the two trailing nucleus or the last nucleus, then both of them will be the male nucleus. Is it clear? So this is the sequence. Vegetative nucleus goes in the front and both the male nucleus follow it. Is it clear? And at this site happens the pollen pistil interaction. After pollen pistil interaction has happened and this pollen tube is growing, so let's just complete this. So it goes through the style and it reaches where? It reaches the ovary, right? And inside the ovary is the ovule, correct? Yes or no? This pollen tube which is developing, it does not enter from here. It goes all the way down and enters from here. Do you understand, everyone? It enters from the down, and this side from where the pollen tube enters into the ovule is known as micropylar end. Is it clear?
Yes.
Yes. So this is the ovary. This is the style. What is this? Inside is the ovule. Now, ovule contains the embryo sac in which the egg cell is present. Okay. Now, this embryo sac is an important structure in angiosperms, and this we have to study in detail. Is this process clear to you? How the pollen tube grows and from where it enters, what is pollen pistil interaction and all of that? What are the nucleuses? How many nucleus are there? What is their position? Everything is clear?
Yes.
Would you like to draw this, everyone?
Yes, sir.
Yes, please draw this and let me know when you're done.
Done, sir.
Nida and Nora,
Yes.
done? Shall I move forward, Nida?
Yes.
Okay. So where does this actual event of fertilization happen? So it happens in the embryo sac. Now, what is an embryo sac? We will study embryo sac of angiosperm. Angiosperms are flowering plants, as you already know. Now, this we are going to study in detail.
Sir, am I audible?
Yes, you're audible. Ask.
Hello.
Tell me. Yes, please tell me.
You were asking what is embryo sac, right?
Yes, I'm saying that we are going to study in detail the embryo sac. Okay. Now, this embryo sac in angiosperms, write down, it is a 7-celled and 8-nucleated structure. Okay. 7-celled and 8-nucleated. What does this mean? It means that if there are only 7 cells and 8 nucleus, then some cell has to have 2 nucleus, correct? If you have to distribute 8 apples among 7 people without dividing the apple, you have to give someone 2 apples. So this is the structure of an embryo sac. Let's draw this. Now in embryo sac, there are it's oval structure and one end. So this is the end from where the pollen tube enters. Remember, pollen tube comes and enters from this this region. This is the developing pollen tube and it enters from here. Okay. Pollen tube entry. So what is this side called? The micropilar end. Clear? And the other side is known as chalazal end. Chalazal, chalazal end. And towards the chalazal end, there are 3 cells which are present. There are these 3 cells which are present with nucleus, each having 1 nucleus. So 3 cells and 3 nucleus, and these 3 cells are called antipodal cells. Okay. So we are done with 3 cells and 3 nucleus. And then towards from where the pollen tube enters, there are some structures known as structures like this, which are called the filiform apparatus. What are they called? They are not cells, but they are called filiform apparatus. Okay. They help in navigation of the pollen tube. What do we mean by navigation of pollen tube? What is navigation? They tell the pollen tube where the egg cell is. Okay. And where is the egg cell exactly? So just beneath the filiform apparatus, there are 2 big cells. Okay. With 2 nucleus. These 2 cells are these 2 cells visible to you? They are called synergid cells. Okay. And behind the synergid cell is the single cell present, which is the egg cell. So they are arranged like this. Okay. So how many cells and how many nucleus are done? 3 and 3, 6. 6 cells and 6 nucleus are done in this embryo sac, right? So we are left with 1 cell which will have 2 nucleus, and that is the centermost cell, which is the biggest cell, known as the central cell. And this cell has 2 nucleus present. Okay. Is it clear, everyone? So this cell is known as the central cell. This is the biggest cell in the embryo sac, and it has 2 nuclei known as polar nuclei. It's called nuclei because it's plural. There are 2 polar nucleus. Is it clear? Mark it outside. So can you count now how many cells and how many nucleus are there? How many cells and how many nucleus?
Seven cells, 8 nucleus.
Yes, so it is a 7-celled, 8 nucleated structure. Is it clear? The overall structure. Now, what happens is this pollen tube is bringing how many male nucleus? The vegetative nucleus does not participate in fertilization. The only nucleus that participates in fertilization is the male nucleus, which is from the generative cell of the pollen tube. Write down. Only the male nuclei, only the male nuclei from the generating cell, from the generative cell of the pollen tube, sorry, of the pollen grain, fertilizes. Okay, so the vegetative nucleus does not fertilize with anything. Now, how many male nuclei are there? Quickly. How many male nuclei are there? People. Aisha, Nida and Noora. How many male nuclei are there?
2.
2, right? So how many fertilization event do you expect? 2, right? Because
Yes.
2 male nucleus, both of them can fertilize with something. But how many egg cells are there in the embryo sac?
3.
Only 1 egg cell is there. Where can you find 3 egg cells? How many egg cells are there? Clearly
1.
1 egg cell. So clearly the first male nucleus, the sorry. So the first male nucleus will go and fuse with the egg cell, right? Is it clear? And what will it give rise to? A male and a female nucleus is fusing. What will become? Zygote, correct? And then this zygote will develop into embryo. Is it clear?
Yes.
What was the ploidy of the male nucleus? Haploid. And the egg cell was also haploid. And the zygote will become diploid, and so will be the embryo. Is it clear? So this is the first fertilization that is happening. So 1 male nucleus is utilized, but there is still another male nucleus left, right? There were 2 male nucleus coming in the pollen tube. So why is there 2 male nucleus? Because there are 2 fertilization events, and the second male nucleus fertilizes with this polar nuclei. So I'm writing it here, the second male nucleus. And what is the ploidy of the male nucleus? Again, it's haploid. It fuses with polar nuclei. How many are there? How many polar nuclei are there? Inside the central cell, how many polar nuclei are there?
2.
Please be vocal. Sorry. Nora.
2.
Right. And what is the ploidy? It's
Both
N.
are haploid.
Yes, N plus N. Both are haploid. We will not write it as 2N because they are not diploid. They are 2 independent nuclei and both are haploid. So I will write it N plus N. Is it clear why I'm writing it N plus N?
Yes.
But in total, how many nucleus are fusing together?
3.
3. So the resultant tissue which is formed is known as endosperm. Is it clear? And the
Yes.
ploidy of endosperm will be triploid, 3N. This is a triploid tissue, and its function is to provide nutrition. It's a nutritive tissue. Okay, nutritive tissue to developing embryo, which was formed here. So do you realize that both of these fertilizations are happening together? That's why this event is known as double fertilization event, and it happens in plants, in angiosperms. Is it clear? What is double fertilization event?
Yes.
Yes. So write down. There are two male nuclei. There are two male nuclei in the pollen tube which enters the embryo sac. In the pollen tube which enters the embryo sac. One male nuclei fuses with the egg cell. One male nuclei fuses with the egg cell and give rise to a zygote. Which later becomes an embryo. And the other male nuclei, the second male nuclei, fuses with the polar nuclei of the central cell. Fuses with the polar nuclei of the central cell to give rise to a triploid tissue called endosperm. Triploid tissue called endosperm. Is it clear? This event is known as double fertilization event. This event is known as double fertilization event. So we understood double fertilization. Is it clear? We also understood embryo sac, right? This is also clear. Now, what is triple fusion? The second fertilization event where the product is endosperm is actually known as a triple fusion. Because can you see that there are in total 3 nucleus fusing together?
Yes.
So this is known as triple fusion event during the double fertilization. Write down. The fusion of a male nucleus with two haploid polar nuclei. The fusion of a male nucleus with two haploid polar nuclei, giving rise to a triploid endosperm, is known as triple fusion. Is known as triple fusion. Is it clear?
Yes.
The third part is also clear, right? Now, quickly draw this structure and write these two fertilization events. They both are happening in the embryo sac. Draw this and let me know.
Then so.
Done?
Yeah.
Okay.
Yes.
Yes. Also, this is written. Is it clear? Is it written? Shall I move forward?
Yes, sir.
Okay. So basically, if you see towards the micropylar end, so this end is important, remember, because where will be the zygote and embryo develop? So one pollen tube will form here. So ultimately this region will become a embryo. So embryo will be here, right? Towards this region, we will have the embryo, right? Yes or no? towards the micropylar end, and towards the chelazal end will be the endosperm, right? This region will become the endosperm later on. Do you understand? And the antipodal cells and the synergid cells both degenerate. Is it clear? They have no use later on. Is it clear?
Yes.
After fertilization, they degenerate. Post fertilization, and they also degenerate post fertilization. So it's easier to understand now that if we divide the developing embryo sac, which develops into embryo, the embryo is towards the micropylar end and the endosperm is towards the chelazal end. Is it clear? Yes.
Yes.
Now in plants, the male pollen grains or the male gametes are referred to as microspores. You know why they are called microspores?
Because they're having lots of spores.
But why the term micro?
They are so
Because they're
tiny.
very small.
Yes, and macro means, oh, sorry, mega means big.
Large.
Humans also, the sperms of the male in humans are very small, so they are called microgametes, and the ovum is called macrogamete or the megagamete. Is it clear? So there is a term in NCERT known as Microsporogenesis. What do you think it is now, if you understand the term Microsporogenesis? Tell me, what is microsporogenesis? Write down. It is the process of formation of pollen grains and pollen grains are also referred to as microspores. Is it clear?
Yes.
If it is microsporogenesis, genesis means
Formation.
yes, so it is the formation of pollen grains from the pollen mother cell, which is known as PMC, pollen mother cell. And whenever we make gametes, through which division gametes are formed? Meiosis or mitosis? Gametes are always formed through meiosis. Okay, reductional division, because gametes are haploid. Okay, now this pollen mother cell is diploid. So PMC is diploid and pollen grains are haploid. Is it clear? So that's why meiosis comes into picture, because it is called reductional division. Make sense? And similarly, if I say what is a megasporogenesis, then megasporogenesis. Quickly.
Formation of megaspores.
Formation of
Megaspores.
what are megaspores?
Eggs.
Ovule, right? Not just the egg cell. That whole embryo sac, the ovule. You understand? That's called, write down. The process of formation of same megaspores, which is ovules. Formation of ovule, just one, okay. From the, and what will be the mother? It will be the MMC from the megaspore mother cell. It's called MMC. Remember MMC and PMC, okay? Again, this will happen through meiosis or mitosis? Through meiosis. All the gametes are made through meiosis. Is it clear? Now, if you know meiosis, if you understand reductional division, tell me quickly, when one cell undergoes meiosis, how many daughter cells are formed? Quickly. Nora? As a result of meiosis, how many daughter cells are formed? Yes, Aisha, you want to answer?
I guess
Mhm.
2.
That is mitosis. A mitosis, which is like a binary fission, forms 2. But in a meiosis, from one cell, when the cycle completes, there are 4 daughter cells. Okay?
Oh, yeah.
And only diploid cells can undergo meiosis, because from one diploid cell, there are 4 haploid cells which are formed. So in case of microsporogenesis, there is no problem. From one pollen mother cell, there are 4 pollen grains that can be formed. Perfect. But in megasporogenesis, it is slightly different. So actually, it just differentiates, like only one MMC is differentiated. So write down, only one out of 4, only one out of 4 daughter cell differentiates into a MMC in the micropylar region. Okay, and then this MMC undergoes meiosis to form 4 megaspores. Write down this the sorry, pen is not working. What was the last line that you wrote, Aisha? The megaspore mother cell, was this the last line? The megaspore mother cell undergoes meiosis. Yes, so write down the MMC undergoes meiosis to form 4 megaspores. Okay, and out of these 4 megaspores, only one becomes functional. One is functional and 3 degenerates. Is it clear?
Yes.
Okay. So do you understand the basic difference between microsporogenesis and megasporogenesis? Both happen through reductional division through meiosis only, but in megasporogenesis, the megaspore mother cell undergoes meiosis and form 4 megaspores, out of which only one is functional and the rest 3 degenerates. Is it clear? Is it clear, everyone?
Yes.
Yeah.
Yes.
Okay, good. Now, so fertilization has happened. Formation of zygote has also happened. We understood how is microspore and megaspore is also formed. That's also clear. Now, what is left? The third stage. What is the third stage? So, first stage is clear. Pollination part is clear. The fertilization or
Yes,
formation of zygote is clear. Now is formation of post-fertilization structure. So write down seeds and fruits are known as post-fertilization structures. Okay. So ultimately, I told you what is future seed. Tell me, which part is called future seed?
Ovule.
Yes, this is future seed. And which part is fruit?
Ovary.
Yes, this is future fruit. Perfect. Now write down the third part that we're going to start. Formation of seed and formation of fruit. Now for that we have to come here and see after this has happened here. So inside here is your this everything is happening in the ovary. Okay. After the fertilization has happened and an embryo is developed, this ovary starts to ripen. What do we mean by ripening? It starts to swell up and starts becoming ripe and big, and this develops into a fruit, and the ovules inside it develops into seed. So you will see many fruits have a single seed. Can you give an example of fruits which have a single seed inside it? Sorry?
Mango.
Mango and peach. Yes, peach has a single seed inside. Yes, and there
Yes.
are and someone was saying something. What else?
Lychee.
Yes, lychee. Perfect. And which fruits have more than one seeds?
Watermelon.
Watermelon. How it has many, like
Papaya.
papaya. Yes, perfect. And guava.
Yes.
Yes. So if a fruit has only one seed, which means it has just one ovule, right, in the ovary. And if someone has multiple seed, it means there were multiple ovules in the ovary. Is it clear or not?
Yes.
So what about strawberry?
Strawberry is not a fruit in real biological terms. We will talk about strawberry. Even apple is not a fruit. You know about, there are two things. One thing is called true fruit and the other is called false fruit. Has it happened in your school? Okay, before you move forward, let me just quickly do one thing. Have I asked you for your exam schedules? When is your school exam happening? Everyone. I don't think I have your schedules here. Okay. Tell me, let's start with Aisha. Aisha, you there?
Yes, sir.
So when is your biology exam?
Sir, we didn't get the dates, but our exams are from 22. And it's like unit test, so it will be over within 1 week.
Okay, so it is 22 June, and I think it's just 1 week. So by the end of June, it will just 7 days, 7 exams kind of thing, right?
Yeah.
So, and what is the syllabus for your biology? You know that?
What we are saying, flowering plants and human reproduction.
Chapter 2 and 3. That's it.
Yeah, and reproductive health also.
Chapter 2, 3, and 4.
Yeah.
That's it.
Yeah.
Okay. And for Nida?
It is on 14th June,
14th June. Okay.
chapter 2 and 3.
No chapter 4.
No,
Okay, just 2 and 3. Okay, cool. And for Nora,
Same like Aisha.
we have chapter 2, 3, and 4.
Yes.
And when it will happen?
Same.
You both are in the same school?
Yes.
Same class also?
I wasn't
No.
aware.
No.
She's studying in ISD.
Okay.
Wow.
You both are in the same school. Okay, so 22nd June. You didn't know that, Aisha? Say hello to your schoolmate. Okay, so cool. So we will be able to do it with these, no problem. Okay, I should know this so that everything is covered in time and you get time to revise.
So
And
one more thing I wanted to ask you, that in human reproduction I'm having one, like there is one topic in which I'm having doubt. So like if after class can you explain me? Because I have test tomorrow on that topic.
okay, I'll do it. Okay. So after the class, you have your test. So I'll finish 10 minutes early and take your doubt, and then you go and have test. Okay. So let's come to formation of seed and fruit. Now, talking about seed, you know that seeds are of two types, monocots. There are two types of plant, monocot plant and dicot plant. You know that? Monocots and dicots, people? Not studied class 11th plants, must have studied plants. Classification of plants, monocot and dicot plant. Yes or no? Quickly. Okay, so this is your homework. You have to go back. You have to figure out what is a monocot plant versus a dicot. This is from your junior classes. You should know this. Is it clear?
Yes, sir.
Yes. Now, when we talk about seed, every seed has a basic structure. Okay, so write down structure of seed. Structure of a seed. Anyone has germinated a seed as a hobby or as a part of any science?
Yes.
Yes, which seed you have germinated? Nora.
Mustard.
Mustard. Was it a part of your holiday homework or something in your junior classes?
Yes.
Summer thing, right? They give you germination as an assignment for biology in summers, where you have to get any plant germinated and bring it to the school. Was it like that? Different stages of it?
Yes.
Yeah, nice. So what did you observe in a mustard? What happened when you put mustard seeds? What happened?
The plant came out.
In how many days? Any rough idea?
2, 3.
to 3 days you saw a small plant coming out. And for how long did you germinated that plant?
Around a week.
Around a week. So you could see the first leaf-like structures coming.
Yes.
Yes. Okay. Now mustard, when you will do this, I want you to figure out what is mustard. Is it a monocot plant or a dicot plant?
Monocot.
Yes. So you know that mustard is a monocot. So you know, do you know what is a monocot and what is a dicot?
Yes.
Just tell me. What is a monocot?
Monocot has only one cotyledon, and dicot has two.
Yes. Single cotyledon. And dicots have two cotyledons. The word mono and di means single and double, and cots comes from cotyledons. And would you like to tell me what are cotyledons? Have you ever germinated any dicot seed? For example, the example which Nora gave, pea, right? Mustard,
Chickpea.
rice, wheat, they are all monocots. Okay. And what are dicots? Pea, perfect. Gram. Sing gram.
Yes.
And pulses also, or bean. They are dicots. Now, what is a cotyledon? For that, we have to, let's see here. So let's take an example of a bean seed. So if you see a bean seed, it is like, let's take a kidney bean. It's like this, right? The seed is like this. Kidney bean,
Yes.
or the one we call rajma, correct? And if you see it from the front, you can see that there is a slight thing, and you can open the seed into two. Yes?
Yes.
The seed can get open into two parts. Or I should not draw it like this. I should draw it. So when you open the seed, it looks something like this as a pair, right? It gets split into two, correct? These two parts are called cotyledons. And what do cotyledons carry? So what I'm going to draw with this part. So what is this part? What is it filled with? What does cotyledons carry inside? What does it contain? Contain endosperm. Is it clear?
Yes.
Seed. And where is the embryo? Here. So if you see carefully, the embryo is in a dicot seed is here. It's in the end. It's sandwiched between the two cotyledons, and it runs through the entire length of the seed. Yes.
Yes.
This part is the embryo. And on the top of the seed, there is a seed coat for any seed, like it's gram or kidney bean or any bean seed. There is a seed coat, right? In a roasted chana or gram, if you have ever had chana, you will feel that there is a seed coat that you remove, and then from the inside is the soft part which we eat, right? That brown covering that we remove on the chana. Yes or no? So
Yes.
technically, every seed has these three parts. It has a seed coat, it has a endosperm, and it has a embryo. Seed coat to protect the seed, endosperm to provide nutrition, and embryo, which is the main part, which is developing inside the seed, and endosperm provide the nutrition to the developing embryo. Now, bean seed is a dicot, so it has two cotyledons. Okay. Similarly, if you see a corn in your monocot also comes corn, right?
Yes.
Corn is also a monocot. So if you see corn or if you see rice, you cannot split that seed into two, right? So it has just one cotyledon, which cannot be open into two. You can break it into two parts, but you cannot open it into two cotyledons. So write down structure of seed. Let's write it here. Write down every seed has three basic structures. First is known as seed coat. Let's start with the seed coat. What do you think seed coat does?
Protection.
One very fundamental thing is it protects, write down, it protects the internal structures of the seed, which also includes embryo. Apart from protection, what does the seed coat does? What do you think? Just protection? Seed coat is a tough covering, right? Yes or no?
Yes.
Write down, seed coat is a rigid protective covering which has two layers. Every seed coat has two layers. You know what are these two layers?
Testa and tegmen.
Yes. And which one is outer layer?
Testa.
Yes. This is the outer layer of the seed coat, and it is also the thick layer. It's thicker comparatively. And tegmen is the inner layer, which is thin as compared to testa. Now, what do you think these two layers are for? Apart from protection, what else a seed coat is doing? How does, you all must have seen seeds in your home. There are many seeds which are stored, right? Your grains are seed, corns are seed, beans, dals, they are all seeds which are stored. If you just store seeds, for example, gram seeds are there, when do they start germinating? If you keep it in a plastic container, do they start germinating?
No.
Why are they not germinating?
Because they become dry.
So what happens? So, but from inside, if a seed is alive, it's a living part. From the inside, it is not dry. No, is it also dry from the inside?
No.
Because if it becomes dry from the inside, then it is dead, right? Because on the inside there is an embryo which is live, there is endosperm which is live, and these are live cells. So if they are living cells, they will contain cytoplasm, correct?
Yes.
And cytoplasm is 95% water, which means every cell has a lot of water stored in it. So inside the seed is moist, it has moisture, it has water. So this seed coat also prevents the seed from dehydrating. Okay, so write down. It prevents the loss of water from inside the seed. So the endosperm and embryo, the cells do not lose water. And it also prevents entry of what we say, parasites or insects from outside. So it is like a wall. It does not allow a lot of water, so it prevents from dehydration of the embryo and endosperm. and it also prevents from entry of insects and other parasites in the seed from outside. Is it clear? Yes or no? But still, the question is not answered. Why the seed does not germinate when it is just kept in a dry plastic container, but when we put it in the soil, it starts germinating? What happens when we put a seed in the soil versus when it is outside? Tell me.
It becomes inactive when it is stored.
So when it is outside, it is dormant, right? That's called seed dormancy. Okay. What is dormancy means? The inactive state where it will not germinate, the embryo will not germinate. So what gives the signal to the embryo to germinate? You know, it's the breaking of the seed coat. Okay. And you know how does seed coat breaks? For this, you have to go to the class 11th concept. You have studied osmosis in class 11th, remember? Movement of water from their high concentration gradient to the low concentration gradient. Yes.
Yes.
And seed coat is dry, right? It does not have moisture in itself. So when you put a seed in the soil, what is the next thing you do? Any germination, any seed, when we put in the soil for germination, what is the next thing we do? We water that seed, right? Yes or no?
Yes.
And the presence of water in the surrounding is a sign of favorable environment, correct? And these seed coats, they start imbibing water. You must have heard of a concept, or you also have studied this in your class 11. The concept is called imbibition. Write it down. This is your second homework. You have to go and figure out what is imbibition. If you remember, you can answer now. Anyone remembers what is imbibition? Imbibition is where the solid substances soak water and they become, the pressure of that water inside starts growing and they break in the effect of hydrostatic pressure. For example, wood when it absorbs water, it starts swelling. You understand? Yes or no? Say yes or no if it is
Yes.
clear. Yes. So seed coat is also like that. It starts imbibing water and then it's break. The moment seed coat breaks, it gives a signal to the embryo to now start germinating out. It's time to come out, rise and shine, because this environment is very favorable. But till the time there is no moisture around, and if you are storing seeds, they always say whenever you are storing any kind of seed, store it dry, right? There should not be moisture or water around the seed. Otherwise, they start germinating. And you have done it in your homes. How many of you have made sprouted chana or sprouted dal in your home? You have made sprouts? No one has.
Yes.
How do you make sprouts? You take chana, you soak it in wet cotton overnight, yes? Or in water overnight,
Yes.
yes? And the next morning when you wake up, you see these small whitish things coming out as tails from the seed, right?
Yes.
That is called sprout. So what happened overnight? Overnight, when the water is imbibed by the seed coat, it breaks and it tells the embryo to start growing. So that is the cue. So write down, seed coat prevents the germination of the embryo. The germination of embryo under unfavorable condition. So till there is dry, unfavorable environment around the seed coat will not break and it will not allow the embryo to come out. Even if the embryo wants to come out, it cannot come out. Okay. It only allows the germination to happen when the conditions are favorable, and presence of water and moisture around is one sign of a favorable condition when you are in the soil or you are near a wet cotton or in water. Is it clear, everyone?
Yes.
So all the functions of seed coat is clear to you. Yes. Right, on the second part of the seed is endosperm, and you already know about endosperm. Write down, it is a triploid tissue. It is triploid tissue. How does triploid tissue helps? If something is triploid, what does it mean? Quickly.
It has 3 nucleus.
No, it does not have 3 nucleus. It has just 1 nucleus. It has a one big nucleus containing 3 copies of the genome. 1 copy came from the male nucleus, 1 copy came from one of the polar nucleus, and the other copy came from the other polar nucleus. Ultimately, they all fuse together. So do not think that a triploid tissue has 3 nucleus. That is called multinucleated tissue. There's a difference between a triploid tissue and a multinucleated tissue. Our muscles are multinucleated. You know that?
Yes.
But each nucleus is diploid. Does it make sense, everyone? Nida, Aisha, and Nora.
Yes.
Yes. So endosperm do not have 3 nucleus. It has 1 single nucleus with 3 copies of the genome, which means three times the DNA material. Okay. The word
Okay.
ploidy comes from copy number of genome. If it is 3n, it has 3 copies. If it is 4n, it has 4 copies. It's tetraploid. If it has 2n, it has 2 copies, diploid. Humans are diploid. All our cells are diploid because we have 2 copies. 1 copy came from the mother and the other copy came from father, sperm and ovum. Do you understand?
Yes.
In plants also, the embryo is diploid because 1 copy came from egg cell and the other copy came from male gamete. Both are haploid, containing one copy each. They fuse together to make 2 copies together. So that's diploid. Triploid means 3 copies, not 3 nucleus. Is it clear, Nora? Crystal clear. So clearly, if it is a triploid tissue, it has more DNA.
Yes.
How does more amount of DNA helps?
Nutrients.
What happens from DNA? What is formed from DNA?
Gene.
DNA makes RNA, right? The gene is transcribed from DNA to RNA, and then RNA makes proteins. Remember this part? DNA to RNA, RNA to proteins. So if it has more DNA, ultimately it will have more protein in the cell. Yes?
Yes.
And more protein means more nutrition. So this cell can also fulfill its own nutritional needs and can also fulfill the nutritional needs of
The embryo.
yes, the developing embryo. Okay. Do you understand how does endosperms help as a nutritive tissue? It has more amount of DNA, so more protein is formed, and more protein means more nutrition, which helps to the embryo to sustain, right? Yes or no? Now write down. Endosperm contains, make this flow chart and write down. Endosperm contains nutrition. Majorly, it contains 2 things. Starch. What is a starch? It's a form of carbohydrate, correct?
Yes.
And the second thing it contains is protein. Yes? These 2 are present in large quantities. It also contains some amount of lipid and other things, but starch and protein majorly. Now, let's come to a question which was asked in the boards. Now let's take an example of rice. What is rice?
A monocot seed.
Perfect. A monocot seed. And what is rice known for? What does it contain? Why do we eat rice? Rice gives us,
Energy.
every food gives us energy, but it is source of what? Is it a source of?
Carbohydrate.
Yes, it's not a source of protein, right? Rice do contain some protein, but we do not consider it a major source of protein. We consider it a major source of carbohydrate, right? Starch. Yes or no?
Yes.
Let me take an example of another seed, which is pulses, like any dal, or let's say bean. What is bean? Bean is a
Dicot seed.
dicot seed. It is a source of all the dal, pulses, or your rajma is a source of.
Proteins.
Yes, we do not call them a source of carbohydrate, right? They are a source of protein. So what happened that one seed is a source of carbohydrate, other seed is a source of protein. What happened to the carbohydrate of this dicot seed? What happened to the starch component? Because this is the major component, right? The first important component is starch, then it also has proteins. But where did the starch? So rice has some amount of protein. That's true. But bean does not have starch, it has protein. Where did the starch go?
It got used up by the embryo.
Yes. So while the developing when the embryo is in seed itself, it is developing there, right? It is just waiting for the right moment to germinate. Yes or no?
Yes.
Write down the starch in beans and pulses got used up. The starch in the endosperm of beans and pulses got used up in embryo development. Is it clear? Everyone, is it clear?
Yes, sir.
Yes. And now about endosperm, some more important things. Questions which have come. Write down, make a star here. And write down. Is a neat question. You know coconut?
Yes.
What is a coconut? It's a fruit, seed, stem. What is it?
Seed.
It's a seed. Is coconut a seed? Nora?
All three.
All the three. How is it all the three? The coconut that we eat, let's say, the white part. When we break a coconut, we take the, so you know that coconut has a hard covering. Then we break the hard covering. Inside we get the softer part, which we eat, and there inside is the water, right? So that hard covering, what is that?
That water is nucleoside.
We will come to water, but what is the hard part which we break? What is that? I'm talking about a ripe, like a ripe coconut. Raw coconut also comes where we just.
Coat.
Sorry, what is it?
Is it the seed coat?
Yes, perfect. It's the seed coat. And then beneath the seed coat, we are already in the seed. So coconut is a seed, right? Because it starts with the seed coat. And what is present inside the seed coat? Endosperm. Yes or no? So the liquid, the coconut water is nothing but a liquid endosperm. Is it clear?
Yes.
Coconut water is a liquid endosperm. And Aisha used the word free nucleus, right, Aisha? You're correct.
Yes, sir.
Why it is liquid? Because all the nucleus, every triploid nucleus is free flowing in the water. So it's a liquid endosperm. It has free nucleus. Slowly over time, you will see that coconut, when it ripens, the seed, when it starts over time, the water becomes less and it starts solidifying as white substance, right? That is the same thing. That is also the endosperm. It's now solidified. Is it clear?
Yes,
Yes.
sir.
Yes. So clearly coconut is a seed because it has a seed coat and it has endosperm inside. Correct. But if it is a seed, it should be present in a fruit, right? Where is the fruit? Where is the ovary? You, any one of you has had coconut water, the green coconut?
Yeah.
You see a green coconut, it's like, the green coconut is like this, right? Yes. Then we cut from this side and put a straw in it and drink the, so inside is a seed coat and there is water, right? Which is the endosperm. Yes or no? This is the
Yes,
seed
sir.
coat. Everyone understanding what I'm trying to say? But you know this green part, which we, in a ripe coconut, it becomes the brown husk, which we peel off and throw. Yes? Yes or no?
Yes.
That is actually the non-edible part. So the fruit here is non-edible. We cannot eat it because it's husk. When it ripens, it becomes husk. Even when it is raw and it is green, we cannot eat it. We do not eat it. We only eat the seed, which is the endosperm, the liquid endosperm. Is it clear?
Yes, sir.
Yes.
Perfect. Okay, so I think endosperm is clear to you. Let's come to the third and the most important part of the seed, which is the embryo. The embryo inside is a diploid structure. Yes? Yes or no? What will embryo develop into? What will embryo become ultimately? People, what will embryo become? Aisha, yes, you're speaking. Are you saying something, Aisha? Nora, write down. No one is speaking, so let me speak. Embryo is the most important part of the seed. It is diploid. This is diploid and develops into new plant upon germination. Is it clear?
Yes.
And embryo, write down, it has three major parts. The first part is called epicotyle. Epicotyle. Okay. And this epicotyle, write down, it is a small embryonic shoot. Small embryonic shoot. You know, epicotyle is also known as plumule. You must have heard this word, plumule and radicle, right? If I make a seed,
Yes.
if let's say this is a gram seed, this is a gram seed looks like this, right? When you put this seed in water, you see that there is something which starts developing from here, right? And also something that start developing like this, right? White structure. One structure, the one
Yes.
which is called plumule, goes and gives rise to shoot, and the other one which is called radicle gives rise to root. Yes or no? So epicotyle is the plumule. It is a small embryonic shoot, right down, which give rise to entire plant shoot system. Gives rise to entire plant shoot system. And there is something called radicle. This is clear again, right? Plumule and radicle both are clear. Right down. Radicle is small embryonic root. Small embryonic root. And what does this gives rise to? What this gives rise to? This gives rise to the plant root system. Okay. But you know that there are two types of roots. Remember primary and secondary roots? You have studied about primary and secondary roots. Yes, Nora? Yes or no? There is this third structure which is called hypocotyle. It's below the epicotyle. It's between the epicotyle and the radicle. And hypocotyle, write down, this is the first structure. This is the first structure that emerges out from a seed. Okay. And forms the primary root. And you must be knowing that a primary root is just like a straight root, right? In the soil. You understand? It does not have branches. Like secondary roots are very branched. Yes or no? But when a plant grows, it does not have branches either in the shoot or in the root. It's just like it has a primary root which goes deep inside the soil and something which comes out with one or two leaves, right? Like this. You understand, everyone? Nora? Nida and Aisha?
Yes.
Yes. So this is that structure, hypocotyle, which comes out the first and it actually anchors the seed. So when you put a seed, when you sow a seed in the soil, do we take care of its orientation? When we put seed in the soil, do we ever think that from which side the radicle will come out and from which side the plumule will come out? We just put the seed in any orientation. Yes or no? Yes or no?
Yes.
But every time when we do that, we see that the plant, when it grows, the shoot comes up and the root goes down. Yes or no?
Yes.
So that anchoring, that proper orientation and anchoring is first facilitated by this hypocotyle. So what does hypocotyle do? Right down? It anchors the plant. It anchors the plant firmly in the soil. For the proper germination. for the proper germination to occur. Okay, is it clear, everyone?
Yes.
Yes.
These structures of seed coat, endosperm, and the embryo is clear.
Yes.
And there is one more structure to the embryo, which is known as the cotyledon, right? This is also a part of the embryo, which basically contains the endosperm, which we have studied above. Okay, so you can just write down cotyledons contains endosperm and provide nutrition, and you already know about it. Contains endosperm, provides nutrition. And the cotyledons, actually, I was asking this to Nora. Nora, when you, if you, because you have germinated a monocot seed, which is mustard, but have you ever germinated a pea or a gram? Have you ever germinated a dicot? Yes or no?
Yes. Yes.
Yeah, so you will see that these leaf-like structures first come up. So when the plant comes up, it opens into two leaf-like structures, which are not actually leaves. They are the dicots. They are the first leaf-like structures that come up. We think they are leaves, but they are dicot. They are the cotyledons. Okay. So write down cotyledons emerge as tiny leaf-like structures from the seedling. Emerge as first tiny leaf-like structure on a seedling. Basically, they are not leaves. And over time, they vanish. They are gone. Where are they gone? Why are they gone? Because it's used up, right? So if you see a plant, let's say, so this plant actually develops like this. It has, and when it comes out in the shoot part, it has two cotyledons that first look like leaf, but over the time you will see when real leaves come, this part diminishes and goes away. Correct? You understand? Everyone understands or not?
Yes.
Yes. Okay, just write down this thing, the embryo part. Just give me one moment. I will just arrange for your test. So you have your test after the exam, after the class.
Done, sir.
Everyone done? Okay, so let's stop it at here. So we have one thing that I'm giving you as write down your homework for. So the third thing that I'm giving you for homework, you have to do quite some. These are smaller ones. You have to just figure out what is a false fruit. In the next class, we'll finish fruit. False fruit versus true fruit. Let me just tell you, it's easy. Any fruit that develops from the ovary, where the ovary ripens and becomes the fruit, is a true fruit in biology. And anything apart from ovary, okay, the structures that I have taught you above when we were discussing the structure of flower, I told you there is receptacle, there is thalamus, etc. So if they develop, they ripe instead of ovary, and it's called a false fruit. Okay, so will you do this, everyone?
You give the same homework in our 11th class also, I remember.
Did you do that?
Yeah, I still have it.
Okay, perfect. So we'll discuss. Oh, you are in a different batch then, right? Okay. So any
And
doubt?
still, by the way, I have the doubt, but it's like I have a test on that topic tomorrow, so I have doubt in that topic. Can you explain it like after the class is over or now?
Yes. So I'll take it now. Tell me, what is the doubt?
Let me see one. Sir, I have the doubt in this topic, like what we are saying, the parturition one and the lactation one, like it's kind of confusing.
Yes. Parturition is the process of giving birth to the baby. The process of delivery, where the baby is delivered out of the birth canal through the vagina from the womb, which is the uterus of a female. That process is called parturition, and parturition happens because of vigorous contractions of the uterus. Okay, so
So
you want,
like before the water breaks, like before that, it starts to give early contractions, right?
yeah, so there are two things. One is called the fetal ejection reflex, right, that your teacher must have told you about. So the fetal ejection reflex comes from the developing fetus, the embryo, and the placenta. So these are mild contractions. They tell the uterus that the baby now needs to come out, and then the water breaks. The water breaking means the amniotic fluid starts leaking because the membrane around the baby, it breaks and all the liquid starts coming out. That is the water breaking event of a female, a pregnant female, in the last month of the pregnancy. And then there are mild contractions of the uterus happening because of placenta and the fetus itself, which gives rise to a next event, which is secretion of oxytocin from the female tract. And this oxytocin makes the uterus contract vigorously, and that leads to parturition. The baby is expelled out. You understood, Aisha?
Okay, so
Yes. And lactation is the process of formation of milk in the mammary glands of the female during pregnancy. So during pregnancy, the hormones like progesterone, estrogen, relaxin, and all these, they along with prolactin also affects the mammary gland, the breasts of the female, and starts making the human milk, the milk in the mammary glands of the mother, which is known as colostrum. So that is called lactation, because the moment the baby will be born, after that the baby needs nutrition, right? And the source of nutrition
sir,
would be the
my
mother.
teacher was saying the cholesterol is that yellowish milk which is produced the first.
Yes. So human milk is known as colostrum. It is thick yellowish fluid which is produced from the mammary glands of the female. It's not like white milk from cows or buffaloes. Human milk is slightly thicker and it has antibodies, nutrition, everything in it. And it's produced during lactation in a female's mammary gland when she's pregnant. Is it clear?
Yes, sir. So annoyed.
Yes.
Uh, so what? Like it was a question as my homework. So what about the females, like those who get, like what was the question? Let me see. Yeah, what happens like when the female is giving birth at 7 months or 8 months, like when the baby is not totally
Yes,
nourished?
that is called premature delivery.
Yeah.
Basically, every vital organ which is important and everything which is important for the baby as a human develops by the end of second trimester. By the end of 6 months, the framework of the baby is developed. It's there. It's a complete baby. The only thing that happens in the last 2-3 months is that muscle mass develops and the organs become stronger. Every organ strengthens and develops more. But every organ is formed, every organ system is formed right in the before the 7th month. So even if a baby is delivered in the 7th month, which is called, or 8th month, which is called premature delivery, that baby has to be kept in intensive care in an incubator where the baby is allowed to develop with some external nutrition, and after that it can develop normally. So what happens is this fetal ejection reflex or this oxytocin um, is not regulated properly, and it is secreted way before it should have been secreted. So this contractions of the uterus starts early and the baby comes out early. But it's okay, like if the baby gets proper care and if the baby is properly formed, it can develop and then grow into a normal individual. It's not like that premature babies will always suffer in their life. They can also grow into a normal individual. That's not a problem, unless and until the babies has some defect or something. Is it clear, Aisha?
So sir, like if the baby is born at 7 months, so for the complete 2 months to complete the 9 month, the baby will be kept there.
Yes, so I told you that by the 7th month, all the organs normally are formed. The baby will not be missing on something major, but it's the chances of some defects are more in premature babies, but they can also be normal. That's not a issue. They are underweight, of course. They are tiny, they are smaller, they have less weight than a normal baby. Okay.
Yes sir, thank you so much.
Okay, perfect. So everyone, Nida, Aisha, and Nora, I want all of you to have your uh, pen and papers with you. Now I will be screening the question paper for today's test. And
Sir, I just, Nora wasn't aware of that. She left the meeting.
oh, was she not aware?
Yeah, she joined late.
Okay, I will ask someone to make Nora join again. In that case, just one moment. Is my screen still shared? Is it visible to you?
Yes sir.
Can you see the paper, everyone?
Yes sir.
Okay, just one more second. Yes. Okay. Okay, it's already shared. So the screen is shared with you. This is your question paper, and I think Nora needs to be called if she is not aware of the test. Okay, so look at the question paper. You will get half an hour for solving this. Okay, and if you have any doubt, please ask me. So there are 4 questions carrying 2 marks and 4 questions carrying 3 marks. Is it clear? Then Aisha, can you see the question paper?
Yes sir.
Yeah, and you have 30 minutes to solve it. The question paper carries 20 marks. After you are done, write your name, badge, my name, and click the picture of the pages that you have solved and send it to the official WhatsApp number of Learnivio from which you get the invitation links for the class. Okay. And for these tests are for you. I have prepared the question for you. We will discuss it in the next class. So I totally rely on your conscience and your honesty. So you have all the means by which you can cheat, but if you are cheating yourself, not me. Okay. So write the questions the best you can. Treat this exam as like you are sitting in your boards or in your school exam and you are writing answers, and then send those answers to me. Then we can discuss what changes or what betterment can be done in your strategies. Okay. If you copy answers, then you will never know what mistakes you are making. Is it clear, Aisha and Nida? Any doubts? Read all the questions first in first 5 minutes and let me know if you have any doubt in any question. If no, then I will leave you with the question paper to solve the question paper in 30 minutes and submit. Please read the question paper and let me know if you have any doubt. Any doubts, Aisha and Nida?
No, sir.
All the questions are clear to you?
2 marks to 1 are clear, and 3 marks, like, it's clear, but I didn't prepare it right.
See, just read the question twice or thrice if you're, and you will realize that it's a very simple question. All these questions are simple, and 6 out of these 8 questions are from previous year's paper, and they are asked exactly in this language. So, and it's simple, like whatever is the language of the question, it is asking you a simple concept. Just read it and you will be able to solve it. Okay. Write the best answer you can without, so if it's a 2-mark question, try to stick around the, like, to the point. Do not beat around the bush a lot. Write exact words which are important for answering. If it's a 3 marks question, you can elaborate a little, and wherever you can think that you can explain better with the help of a flow chart or a diagram, do that. Though I have not included any diagram related questions specifically, but you can use flow charts to explain certain things if you wish to do that. Okay. Any doubt, Nida?
No.
Okay. So Nida and Aisha, all the best. Please complete the test, and I would encourage, if you attempt all the questions, do not leave questions. And this is a strategy you should follow for your boards and school exams as well. Leaving a question is a sure shot